This is a neuron, which has four main parts. The dendrites receive information. The cell body processes and integrates that information. The axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long, as they often need to transmit information over long distances. As we just saw, the dendrites are the part of the neuron that receives incoming signals. Based on the strength of this incoming stimulation, the neuron must decide whether to pass that signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon in a phenomenon called an action potential. When this happens, we say the neuron fires. Transmission of a neuronal signal is entirely dependent on the movement of ions or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed between the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions. When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside the cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, we also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called an electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately -70 millivolts. This means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to establishing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane and the bulk solution on either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electro gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in the membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require a signal to tell them to open or close. For example, voltage-gated channels only open when the membrane potential reaches a certain value. On the other hand, ligand-gated ion channels are triggered to open when they are bound by a specific molecule. Mechanically gated ion channels open in response to physical forces, such as changes in length or changes in pressure. Ion channels, for example, typically only allow a single ion.
All right. So this is basically about the action potential that we did. Okay. So all the ions that we mentioned, they have their unique gates, and most of their gates are voltage gates, which allows only that ion to pass through. So that's what we normally say it is the cell is semi-permeable because it allows only that ion to pass through the gate. So sodium, for instance, it has its own gate, which allows only sodium to pass through. And the same thing applies to all the other ions. Okay, so let's watch. I think this video centers around only action potential, what we did. So the video that I want you to watch is what? Is less than 2 minutes. That's synaptic transmission. So one thing is the synaptic transmission happens at the terminal button. So after action potential has taken place at the axon, then it will move to the terminal button. Then the terminal button will then release that sort of information. It is more or less like a chemical. It will release it to another neuron, its dendrite. So it means the first neuron would release the information at its terminal button and move it to the second neuron, it's what? Dendrite. That's what happens. So let's watch this video.
2-minute neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss synaptic transmission. Most communication between neurons occurs at a specialized structure called a synapse. A synapse is an area where two neurons come close enough to one another that they are able to pass chemical signals from one cell to another. The neurons are not actually connected, but are separated by a microscopically small space called the synaptic cleft. The cleft is less than 40 nanometers wide. By comparison, a human hair is about 75,000 nanometers wide. The neuron where the signal is initiated is called the presynaptic neuron, while the neuron that receives the signal is called the postsynaptic neuron. In the presynaptic neuron, there are chemical signals called neurotransmitters that are packaged into small sacs called vesicles.
Good.
Each vesicle can contain thousands of neurotransmitters.
Okay, so let me explain this. So this is the first neuron that I'm talking about, neuron A. And this is neuron B. So neuron A, okay, let's say there's the axon. So the axon has what? Is transmitting the information. Then it will get to this side, which is the terminal button. So this is the terminal button. These areas are the terminal button, or this structure is the terminal button. So the terminal button will then what? Will then release the information to another neuron, the receptor of another neuron. So this neuron, the first site that will receive that sort of information is what? Is the dendrite. So it means when it comes to neuron A, it is a terminal button that releases the information. Then neuron B will receive that sort of information, and the part that receives the information when it comes to neuron B is what? Is the dendrite. Okay. So when this happens, let me erase everything. So the information I'm talking about, they are more or less like vesicles. This is the vesicle that they are saying. So it means action potential, the action potential that we are saying, they are transporting something. Like, they are more or less like a chemical being transported by the axon. The axon is transporting what? A chemical. And the chemical the axon is transporting is what? Is this vesicle. So it will be transported to where? To the terminal button. So let's say there's a vesicle in here. So there's the axon. The two horizontal lines I've drawn is maybe the axon, and the axon is transporting the vesicle, the circles that I'm drawing. Okay, and these vesicles, they contain some chemicals. They contain some chemicals known as neurotransmitters. Okay, they contain some chemicals known as neurotransmitters. So the moment the vesicle gets to the base of what? Of the terminal button, it gets to the base. So if you see that this green one, this green vesicle just got to the base, it will then release the neurotransmitter. Then the neurotransmitter will fill this receptor. It will fill this side, these receptors. Then it will cause an action. So when the neurotransmitters gets to the neuron B, the dendrite of neuron B, it will cause a reaction. It will cause either one of these reactions. It will cause one of these reactions. It would either excite or inhibit neuron B. So basically that's what it is all about, the synaptic transmission. So the synaptic transmission, after action, in the course of action potential taking place, it means action potential, they are carrying what? Some information, that's vesicles. They are transporting vesicles within the axon or along the axon. And these vesicles, they contain neurotransmitters. So the moment the vesicle gets to the base of what? Of the terminal button, or sometimes in books they call it the presynaptic. So the terminal button is also known as the presynaptic membrane. And neuron B is also known as postsynaptic membrane. So it will then, I'm continuing. The moment this thing, the terminal, the vesicle gets to the base, it will then release it to what? To neuron B. And interestingly, you could see that there's a gap, there's a space around the side. There's a space here, this is the space. We call that space a synapse. They are very, very small. You can't see it with your eyes. So mostly they use what? It is kind of a microscopic space. And we call that space synapse or synaptic cleft. We call it synapse, the space or the gap between the two neurons. We call it synapse or synaptic. So the moment I'm explaining again, so the moment the vesicles gets to the base, the vesicle contains these neurotransmitters like this. So the moment it gets to the base, it will cause an action so that it will then release the neurotransmitter along the synapse, that's the gaps or the space. Then the neurotransmitter will fill which side? It will fill the receptors here, the receptors of what? Of neuron B. So basically that's how the reaction is all about. So the moment it excites on the receptors, then it will either excite this neuron or inhibit this neuron. So when it excites this neuron B, we then call it, let me type this, we call it excitatory postsynaptic potential. So when it excites neuron B, we call it excitatory postsynaptic potential. But when it inhibits neuron B, we call it inhibitory postsynaptic potential. Okay, so please, for now, any question? If you have a question, kindly raise up your hand. Okay, so I can go ahead. All right, so nobody raised up his or her hand, so I assume that it is self-explanatory, so I can move on. So one thing is, when it excites like this, basically what we are saying is that it will then, for us to say that it is leading to excitatory postsynaptic potential, then it means that it will cause this neuron B to also what? Fire, or cause it to also go through an action potential. That's what we mean by excitatory postsynaptic potential. So it means it increases the activity of what? Of neuron B. That's an action potential takes place within neuron B when a neurotransmitter sites on what? On the receptor of neuron B, and it causes excitatory postsynaptic potential. What it means is that it causes an action potential within neuron B. But when we say there's an inhibitory postsynaptic potential within neuron B, then it means that it then what? It then decreases the activity or whatever activity that was going on within what? Within neuron B. So naturally, that's what happens. And we have several neurotransmitters and its function. Some neurotransmitters, they excite, others what? Inhibit. So depending on the type of neurotransmitter, that would be what? That would lie on the receptor of neuron B. It will help us know whether it will excite or inhibit. So currently, that's what it is all about. Zoom just alerted me. I have less than 10 minutes. So let me go back to your slides so that you know some of the neurotransmitters. Okay, Randy, go ahead.
Yes, sir. Good afternoon, sir.
Good afternoon.
With all these actions, there's synaptic transmissions that you were explaining. Do we have control over these things? Do we have control over them?
Yes. So, for instance, when you are happy, for instance, okay, it means that there's a neurotransmitter making you excited. So there's an activity going on within your brain. Okay. That is causing such thing to happen to you.
Okay.
Yes. So it means whatever activities that is going on within ourselves, it is based on the neurotransmitter. So when someone is depressed, it means there's a neurotransmitter happening, a chemical causing that.
Right.
Yeah.
So it means you can actually you can have, when you are trained, you can control your mood. That's how come you are able to control your mood, whether you should be happy, whether you should be sad, and all that. Correct?
Yeah. Because, so for instance, if someone is sad and we want, so for instance, if someone is sad, we normally say that it means there's a neurotransmitter known as serotonin. That's the happy neurotransmitter. It should be quite moderate within your system. So if someone is sad, it means the person's serotonin level is what? It's low. No, it's low. Yes. So this thing is low within a person's system.
Right.
So what we normally do is that we have certain things, we help the person so that out of it, the person will begin to produce some little serotonin that will help him or her to what? To be in a stabilized mood or become a bit happy.
Roger that.
So that's what happens. So we normally, yes. So we have several of them, and these are the main classes of neurotransmitters you are supposed to know.
Okay.
Okay, then under amino acid, we have glutamate. Glutamate serves as an excitatory neurotransmitter. So in exams, they can ask you, mention any excitatory neurotransmitter. Glutamate is an excitatory neurotransmitter. So it means if glutamate is the one coming, it means it is coming over to excite the other neuron for it to cause an action potential.
Right.
GABA is an inhibitory neurotransmitter. The full name of GABA is gamma amino butyric acid. GABA. It is an inhibitory neurotransmitter. Then acetylcholine, which is this, is an excitatory neurotransmitter. Then serotonin is an inhibitory neurotransmitter. Dopamine, it serves as both inhibitory and excitatory, dopamine. Then norepinephrine is an excitatory neurotransmitter. So these are some of the things you need to know. And I would advise you that you should know the function of each. We have different function of it. So I would explain a few of them. You have to do research on your own. So for instance, glutamate. It plays a significant role in memory and learning. Glutamate, it plays a significant role in memory and learning. Then with GABA, GABA in most situations, it's what? It decreases the activity of what? Of the brain. It decreases. So that's why we're saying it is an inhibitory. It decreases the activity that goes on within the brain. So if someone have seizures, I know most of you know what seizures is. An example is epilepsy. The person has epilepsy because there are a lot of activities going on within the brain. The brain is firing a lot. There's a lot of firings going on within the brain. So in most situations, drugs which are given to epileptic patients when they go through that seizures, it contains GABA so that it would decrease the firings going on in the brain. All right, then acetylcholine also plays a significant role when it comes to memory. It is also an excitatory neurotransmitter. And in most situations, we normally say that when acetylcholine is low within someone's system, it could lead to a disorder known as Alzheimer's. That means the person will have memory loss, Alzheimer's, or dementia, Alzheimer's. The A is capital, disease, Alzheimer's disease. Okay, so I have less than 5 minutes, so let me finish this. Then serotonin, it plays a significant role when it comes to mood. Yes, then with dopamine, it's also, we normally say that, okay, with serotonin, I've already indicated. So when the person has low levels of serotonin, it means that the person will be what? To be sad. All right, then with dopamine, in most situations, we normally say it is an excitatory neurotransmitter, and lower levels of dopamine, lower levels of dopamine could lead to a disorder known as Parkinson's disease. And higher level of dopamine leads to a disorder known as schizophrenia. All right, that's all. Then the last one I'll discuss is norepinephrine. Also, sometimes we call it noradrenaline or adrenaline. So norepinephrine also plays a significant role when it comes to mood. And higher levels of norepinephrine could lead to anxiety. All right, so I have less than 2 minutes, then it will cut. If you have, I would take about 1 or 2 questions. So if you have any question, kindly raise up your hand. Please be snappy about this. I have less than 2 minutes. That's 1 hour, 30 minutes. So if you have a question, okay, Albert, so please go ahead. Albert, kindly unmute yourself and speak.
Hello, sir.
Go ahead.
Hello, sir.
Please go ahead, Albert.
Okay, sir, please, the full name of Gaba again. No, the line wasn't clear, so I didn't hear it.
I said it's gamma amino butyric acid.
Gaba amino.
Is there butyric acid?
Okay, sir. I've seen it. Thank you.
All right. So if there's no question, then that'll be there. Okay, Samuel, please be snappy. I have less than a minute.
Yeah, please, with the amino acid, are they medication that have been given to the people, or?
They are natural neurotransmitters within our system, but we'll talk more about it next week. There are some drugs that are made for, that works like neurotransmitters. So most of the drugs that goes on in our system, it has been made in such a way that it has some component of what? Of neurotransmitters.